Very nice. So yeah, thanks again. And I already apologize because I think that my presentation is not going to be as colorful and nice as the one from Claudia. But anyway. So yeah, as Mike said, this is part of my PhD project in which I'm developing it at the CPO, which is part of the University of Porto, in collaboration with the Institute of Biomedicine of Valencia and the University of Bangor. And basically, in my project, I'm studying I'm characterizing the venom of Western Mediterranean vipers and studying its variation. So now I will talk about what has been basically my first project and the first species that I studied, which is Lataste viper. And it's over. So I think it's quite definitive. The results that we present today are quite definitive. So first of all, a bit of background, just to give you an idea of what we're going to talk about. What is snake venom? Snake venom has been basically considered as a complex cocktail of the active compounds, which are mainly proteins and peptides, which of course are able to affect the homeostasis of the envenomated organism. And so far, more than 60 different snake venom protein families have been discovered, and they are known to affect different systems of a living being, mainly cardiovascular, neuromuscular, and the hemostatic. And it has also been demonstrated that the composition of snake venom varies depending on different factors, like, for example, age, sex, phylogenetic relationships, and especially diet, which seems to be the main driver of venom variation. So the species I analyzed for this work is Lataste viper, that you can see here. It's a decently sized old world viper, and it is endemic to the Iberian Peninsula. And mainly it's reptiles and small mammals, although it's known to be quite generalist. In fact, it's occasionally invertebrates, amphibians, and birds. And the venom, although considered of medical relevance by the World Health Organization, is so far poorly studied, although some coagulative effects have been reported. What is interesting is that for the ecology of this viper, it has been described an ontogenetic dietary shift, in the sense that juveniles appear to mainly prey on ectotherms, especially small lizards, while adults mainly prey on endotherms, like especially rodents and micromammals in general. And in this graph also we can see that, for example, the larger the snake, the larger the prey they try to ingest, which is of course a general pattern that we see in snakes. So the aims of the study are basically the characterization of the Vipera lataste venom proteome, which is so far lacking. The investigation of the presence of an ontogenetic shift in the composition of Vipera lataste venom that could eventually mirror the ontogenetic shift in diet. And also, finally, to assess the presence of geographic variation between the venoms exclusively of adult vipers from different locations. For this work, I've been sampling vipers in 2 areas of northern Portugal, 2 very well-known populations, which are defined as Boscá and Jerez. You can see here on the map. Why did we select these 2 areas? Basically because they host very abundant viper populations, therefore sampling was decently easy. And the vipers from both populations belong to the same mitochondrial haplotype. So basically there will be no genetic influence on the eventual differences seen in the venom. And the 2 localities sampled are environmentally different, in the sense that, as you can see here, the population 1, which is Boscá, is clearly a coastal environment, where the climate is more or less mild for the whole year, and the disturbance is present all year round, in the sense that very close there are like cultivated fields and crop fields. While in the second case, in the case of population 2, which is Jerez, it is a clearly mountain environment. The climate is colder and, let's say, harsher for the whole year. And the disturbance is mostly seasonal, in the sense that it's mainly touristic, so it is focused on specific months of the year. The methodology applied here is the method of Venomics, which is basically the analysis of the snake venom through reverse phase high product lipid chromatography, with the production of the chromatograms that you see here in the second picture. Then eventually there is the electrophoresis and the digestions of the bands that we get by electrophoresis of the venom. Eventually, the use of mass spectrometry. And in the end, the single peptides are searched through online databases. And finally, we get the pie chart, which constitutes the proteome and represents the relative abundances of the toxin families present in the venom. Also, we applied an individual-based approach in the sense that we analyzed all the peaks present in each chromatogram that we produced, but excluding those that had frequency equal to 1. Here there is an example of the matrix that we used, meaning that basically for each specimen, we recorded the peaks that were present in the venom chromatogram. This is just a little example. And then we applied non-metric multidimensional scaling, in short NMDS, which is basically a way to represent the association and the similarities between the venom profiles. And then we applied regression models to investigate the relationship between the NMDS scores that we got and that we use as a response variable, and some characteristics that we got from the vipers, like snout main length, sex, date of collection, and population of origin. Talking about results, in total we had 54 individual venoms, more or less, let's say almost 50-50 between the two populations, but slightly more from Villachat. And we also had 2 pools of juvenile venoms. And here we can see basically that we can already see by eye some differences in the chromatograms. For example, you can see that the juveniles, the main differences here are around -15 and 17. You can see that some adults present a very clear peak, while juveniles tend not to have it. And conversely, juveniles tend to have very high late chromatogram peaks, while the adults tend to have them in a lower number and with lower intensity. So based on these differences and on the NMDS analysis, we got this ordination plot. In blue, you can see the venoms belonging to the population of Villachat, and in red, you can see the venoms relative to the venoms of Jerez. The triangles represent the juveniles. You can see that they are quite extreme compared to their position is clearly different from the ones of the adults, meaning that there is indeed some kind of difference in the number of peaks. And so you can see that the two populations are quite different, although with some level of overlapping. And among these venoms, we selected one juvenile and 2 adults per population, and we produced the proteomes. And as you can see, 4 toxin families compose more than 50% of each venom proteome. These families are specifically the C-type lectins, phospholipases A2, snake venom metalloproteinases, and snake venom serine proteinases. What we can see here is that there are clearly some ontogenetic differences, in the sense that the proteomes A and B belong to 2 juveniles, and although the 2 juveniles were from 2 different populations, their compositions are extremely similar. When on the contrary, we can see that the proteomes of specimens C, D, and E, that are adults, are also quite similar, although between them, but very different from the juveniles in terms of composition. Finally, we can see that the venom that here we defined as F, belonging to sample 19VL029, was likely sampled at a pivotal stage between adult and juvenile. In fact, we can see that there are some characteristics that are kind of juvenile-like, like the great abundance of metalloproteinases, but also some characteristics that are adult-like a great abundance of phospholipases A2. The major conclusions of this work are that we found up to 22 different classes of venom components in Vipera Lataste venom. So a quite decent compositional diversity, which we suspect might have developed to effectively target different prey physiologies, being Vipera Lataste a very generalistic predator. Also, we found clear evidence of ontogenetic shift in venom, the same as reported in other viper species, also from the nootropics, in the sense that juveniles have a metalloproteinases-rich venom, which is supposed to help a quick incapacitation of the prey, while adults have venoms that are rich in serine proteases and phospholipases A2, which supposedly might help in the digestion of mammalian prey. But still, this has to be clearly investigated. And finally, we found that the geographic variation was supported by our statistical analysis, in the sense that among all the predictors that we tested, only the population of origin seemed to add a significant effect on the NMDS scores that we got. Therefore, we suspect that the differences in venom composition are likely due to different local selective regimes other than to other factors. Here there are some references in case you are interested. And finally, I want to acknowledge my three supervisors, which are Dr. Fernando Martinez Freire, Wolfgang Wuster, and Juan Calvete, and all the other people that helped me in the development of this work. And thank you very much for your attention.
Thank you, Ignazio. So if anybody has a question, now's the time for it. There's some applause. And even more. If I saw it correctly, you had in one of the venoms, I think in the first pie chart, shown some disintegrants with a more higher percentage, but a low number of class 2 metalloproteinase. Do you have an explanation for this?
I can get better at it. Sorry. Do you see it?
In the first pie chart you showed.
Okay, so A, right?
No, on the other slide. Or wasn't it the latastei?
Ah, sorry, you mean, wait. Will it be which? This one?
This is just the general proteome and not from the Vipera.
No.
Ah, okay.
Yeah, I think this is rattlesnake. I don't remember.
Okay,
exactly.
then totally ignore my question.
Okay. Fine.
Okay, so there are no further questions. Then I think
I have one.
There's one. Okay.
Hi, Ignazio.
Hola, Gabri.
So I was wondering, like it's more like a general question that with the 3 vipers that you're working, once you have established the venom composition of the 3 species, no, of the Iberian Peninsula, do you think that you can infer information about hybrids or specimens that could be in progress between the species just with the venom composition?
Hybrids. Well, that's a very good question. I think that so there are some studies that have been developed on hybrid specimens, and what they found out is that in the case of two parental species having very different or quite stark different compositions in venom, the hybrids are something that is halfway between like they get characters of both parental species, much like morphology, I would say, or something like that. I don't know if this could be the case, because of course there are hybrids, for example, of Vipera lataste and Vipera aspis in northern Spain. But in the current state of the things, I'm really unable to tell you, because I also suspect that the composition could be quite similar, in the sense that these parental species usually stay in are probably competitors also for the same resources. Therefore, if we assume that the venom has been developing especially to target prey physiology, if they feed on the same prey, I don't expect too many differences between the two parental species. But this is pure speculation, because I have no idea of what the Vipera in northern Spain, the proteome of the Vipera in northern Spain could be.
Yeah, for me it was that out of curiosity. It was really nice to talk. I'm glad.
Thank you.